Well, I want to thank everybody for coming again once to our session, a virtual live INTERPHEX live session. As most of you know, this would normally be taking place at the Crystal Palace at the Javits Center in New York City. Well, we don't have to go into the biology and the pandemic discussion to understand why this is virtual. However, the folks at INTERPHEX and Reed Exhibit felt it strongly enough that these panel sessions go forward, none the least of which is that they become a resource for our colleagues out there. And I have the privilege once again to introduce our panel. Our panelists is Hillel Cohen from Executive Director of Scientific Affairs at Sandoz, Rick Morris, Senior VP at Pall Biotech. Welcome, guys. And of course, my old friend Mike Ultee, CEO, owner of Ulteemit Consulting. So what we're going to do is right now, Mike and I had a brief discussion, and I thought what we're going to do, Mike, is if we could lead off that this year is the 10th anniversary of the Biologic Price Competition Innovation Act. And that began the movement that we all sit here and talk about. So why isn't the whole world out filled up with biosimilars, Mike?
Okay, well, there's three main reasons, as I see it. First is regulatory uncertainties. There was a lot of initial uncertainty after the act was signed 10 years ago about, where this was going, and it took 2 years before FDA came out with guidance, the first guidance documents that would address the uncertainties, because the big question is how similar did you have to be to a reference or an innovator product in order to get licensure as a biosimilar. So the first guidance is issued 2 years after the act were part of the delay, but these guidances answered some questions but raised many others. And so since then, there have been many additional guidances issued by FDA to clarify, and even some guidances retracted as newer guidance came forth to reflect their current thinking. And these continue to come out even this year, several. And some have been focused on general principles, some on analytical considerations, and other specific topic areas. And these have been very helpful. And over the 8 years that they've been coming out, they've really kind of cleaned up the regulatory uncertainty. So now there's a pretty clear path on what a sponsor needs to do. But this was all part of the delay compared to, say, other parts of the world like Europe, which which is way ahead. But it's principally ahead because the U.S. started much later. The FDA guidance came in 2012, 9 years after the European guidance was issued. So, but in the first 4 years of the EU initial guidance and the FDA guidance, a similar number of products were approved, 16 in one case, 13 in another. So the U.S., as I said, just started a bit late compared to the EU. Two other reasons for the slowness is that there's been a lot of patent litigation between the holder of the brand name or reference product and those trying to develop biosimilars. And many of the patents around a biological product are very complex and multitudinous. In fact, patent attorneys call them a patent thicket, which means there's just a giant jumble of them, as many as 100 patents around a complex biologic. And this can result in extensive litigation, which has delayed market entry and usually ends up with a settlement which between the reference product holder and the biosimilar company, which agreeing to delay market entry for some years. And finally, the patient populations for many of the biologics that are approved are for rare diseases where patient numbers are much less. So when you start going into multiple products for that indication, the attractiveness of the market diminishes. But those, I think, are the big reasons for this kind of slow uptake. But in the last year, we've seen a lot of progress, a lot of new approvals, and I think going forward, we're going to see a much more rapid pace in the U.S. on biosimilars.
I'd like to add an additional thought to what Michael said. So obviously I agree with what you've said, but there are also some commercial barriers as well, where the reference product holder, the licensed product, reference products company will have various bundling packages, rebates. So, I mean, this is really not my area of expertise, but I'm aware that it happens. So there are significant commercial barriers. And there are also some perverse incentives as well. When you get reimbursed at a certain percentage of the product, there's actually a perverse incentive to prescribe a more expensive product. And we see that happening. So there are efforts being thought of in the halls of Congress and in the state houses to try and put some incentives into place. So I think that's another major. And of course, as Michael alluded to, the physicians and patients have to become more comfortable with the biosimilars as well, so that it become more accepted.
What's interesting, Hillel, it's odd, it's good that you bring this up. And just as an aside, the panel discussion we had on patient-centric supply chain with Dr. Wayne Weingarten, he works at the Pacific Research Institute, and he addressed the biosimilars market and reimbursement quandary that's ongoing, and he confirms everything that you had said. That's a different session that we recorded, but yeah, it would synergize excellent with the conversation. Same thing. It's almost counterintuitive. So, but let me ask this question then. So is there actually prevention? Is there anti-competitive action ongoing? I mean, I could see there's the reimbursement, but there's also are people building in anti-competitive exercises? All right, Hillel, you want to lead off with that?
Well, there's anti-competitive is there's things that a company can do that are completely legal. There's no reason that you can't advertise heavily on the print and television media to people that they should stick to your product. Well, not to stick to your product. The borderline is when you start saying things that are disparaging or are misleading. So that's one aspect. I mean, and we'll talk about that a little bit about the actions of the FDA and FTC. Another thing you can talk about that could be considered as anti-competitive is if you have 150 plus patents around something, is that anti-competitive in its nature? Well, the courts have just ruled that it's actually the company that has this, which is AbbVie, around their Humira product. It's actually illegal to have this many patents and to try to enforce them. So is it anti-competitive? Yes. Is it legal? Also yes. So there are things that companies, the originator companies, can do to protect their market share, which are perfectly fine. I mean, the biosimilar companies at this point are just simply asking for a level playing field. And I think over time, that will happen.
Mm-hmm.
Mike, you want to add to that?
Sure. Should I go into that FTC-FDA collaboration?
Yes.
Yeah, that was announced earlier this year, and in fact, I'll have Hillel comment on that in a moment since he was at the meeting and on a panel there. But basically, the Food and Drug Administration (FDA) and the Federal Trade Commission (FTC) have agreed to a joint effort to help promote biosimilars by clamping down on a number of things like the misinformation campaigns that Hillel alluded to from certain brand name companies that imply or state more explicitly that the licensed biosimilar is somehow inferior to the brand name or reference product. And secondly, the agencies will assist developers of biosimilars in obtaining samples of reference product. This has been a hurdle, sometimes a barrier to entry, because the holders of the company selling that product doesn't really want to give out samples of it. And the FTC will take a closer look at patent settlement cases, which involve a delay to market provision for the biosimilar company. I mentioned these earlier. And for example, AbbVie, maker of Humira, the largest selling pharmaceutical company, product in the U.S., has a number of these agreements in place with major companies, and really it delays a biosimilar for Humira until 2023, even though the patents have expired on Humira because of the number of patents and, of course, the ongoing litigation that AbbVie would engage in should the product be brought to market by the biosimilar developer. Anyway, the Federal Trade Commission is going to take a look at that. And finally, the FDA described a pathway to licensure for a biosimilar for fewer than all the indications of the reference product. This is something that may be necessary when some indications are still covered by patent restrictions, but not all of them. So at this point, I'll defer to Hillel and see what he might like to add to my comments.
Yeah, okay, this was a hearing held on March 9th, what happened to be the very last day that the FDA held meetings. March 10th, they closed down. So it's literally the very last day. Just in time, yes. The FDA actually built an FTC. It's actually very rare to have joint meetings of two agencies in that manner. It happens, but only several times in a decade. So it's really rare. This has been something that's been building up. And if you take a look at almost the agenda built, the first thing it says, they said the first step one is, well, what can the FDA do? What can the FTC do under these circumstances? Then they went to say, well, what have we encountered? What powers of enforcement do they encounter? There was a session on which I was the panel saying, what has been encountered? They had a patient group, they had a healthcare provider system, Kaiser Permanente. The Arthritis Foundation was the patient group, and I was there representing the industry as well. And they said, well, what examples of misinformation have you encountered? There was another set of two lawyers out there who's saying, do these things rise to anti-competitive behavior and antitrust? A professor from Harvard, another one from Rutgers, the law professors. Their conclusions was yes, that it does. So having said all this, now what does the FDA, what does the FTC do? Because they actually have very different enforcement capabilities. And we really haven't seen much yet because, well, COVID-19 will slow things down. But secondly, having essentially gotten the green light to go ahead, have identified the issues, I think it's fair to say that the FDA is continuing their education efforts, albeit now virtually. And the FTC is now seeing what existed, what legal case, if any, can they bring together. Now, one interesting aside that I can tell someone I'm co-chair of the education committee, one of the trade associations. There have been certain websites that have suddenly disappeared from various companies where there were some questionable statements being made that fell in the gray zone or went past the gray zone. And those they're no longer there. It doesn't say that the FTC doesn't have web screen shots of what was there before. I'm assuming that they do. And also, I think it's if companies have, or individuals have come across something they feel is misleading, I think they are encouraged to approach the FDA and FTC. Those are the appropriate people. The FDA and FTC then take that information and they do their own evaluation. Is this a legitimate claim or not? I mean, just because someone complains doesn't mean it rises to the level that something should be done about it. On the other hand, one of the FDA and FTC look at it and they verify it as being a valid situation, valid complaint. In that case, they may want to do something about it. So the meeting was first held March 9th. I wouldn't be surprised if we start seeing some things happen as well. Another thing the people listening to this should be aware of, the FDA just a month prior to this meeting came out with a guidance on promotion of biologics and biosimilars, which is rather unusual, and they were very explicit. Now, they were very clear. They said the biosimilar can't say XY, the originator can't say XYZ. They're very clear about what would be misinformation, what you can say, it can say. So just the fact that that guidance came out just in advance of the meeting means the FDA is also looking at it closely. So I think we can see that twofold. One, actions might be taken, but equally important, companies and various organizations, and it's not just the originator companies, sometimes they're proxies as well. In fact, that's another tactic that we see being used. There are various proxy organizations being used to give the originator message as well. So there's a lot going in that area. And I think we're going to see perhaps a cleaner landscape now that people are aware the FDA and FTC are looking into this.
Let me, as I've listened to we've discussed this a couple of years, and this year seems to be particularly complex as far as issues and so, I mean, all of this goes on, and for the sake of a better term, I'll say it's in the background. But Hillel, let me put it as a firm that's what you do. Let me ask you, what is the barrier for uptake here? I mean, is this stuff all put aside? But if this stuff was put aside, does that help, or what?
Well, that's actually a good question. I mean, a couple things. One, Michael said the barrier used to be regulatory. I think that's much less of an issue. It's not completely resolved yet because we still have issues of interchangeability that the FDA hasn't yet answered yet. But for many things, and many things like the industry is not necessarily happy with the outcome, such as naming, but at least we have an outcome. There is the issue of the litigation. If we have 17 on the market and almost 30 that have been approved, that means close to half of the products approved in the US are not yet marketed. So there are barriers to marketing, and those are really litigation and patent issues. So that's one. And of those that are on the market, the question is, are they being used? Now, there are two major barriers to being used, and we've talked about them. The first are the commercial barriers, rebates, other tools being used, reimbursement challenges of Medicaid, Medicare as well, that sometimes have a perverse incentives of disincentivizing biosimilars. And the third, of course, is getting physicians and healthcare pharmacists and patients comfortable with use of biosimilars. Now, I think we're going to talk about it later. The uptake varies widely among products. For filgrastim, the biosimilar filgrastim, Zarzio, which happens to be my company's product, we're now at over 50% market share. Now, the opposite extreme of the infliximabs are at around 10% market share. You have a lot of products out there that are in the 20, 25% market share. Some have been out for a short period of time, some longer. So there is uptake, maybe not as large as people would have liked. Maybe I think some of these activities are slowing down uptake. But I think so in other words, it comes from a multiplicity of things coming out that are keeping it away. Biggest is litigation, commercial, reimbursements, and then the knowledge and comfort level with use of these products.
Russ, Hillel, I have a question on top of this, if you don't mind. There's like the BLA approvals of like number of products in March as well for biologicals. Will that sort of drive more biological activity, more coming through in the market, or it's just they're there and no one can really do anything?
Well, perhaps you're referring to what some people call the virtuous cycle, which is we're hoping that the greater use of biosimilars will provide savings to the healthcare system so that they can start using some of these novel, innovative, and relatively expensive therapies. They are very expensive therapies. Now, you see the biosimilars are coming to market, besides the list price, the actual price is around a third of that originator. And then we see many of the originators are also lowering their prices as well. But there's a limited amount of money in the healthcare system. And I think we need to create the headspace.
But, we've been talking about this as a team here for several years. So, Mike, I mean, what's the perspective? I mean, compared to 5 years ago, right? I mean, what are the principle? So in other words, here we are as a team. We've talked about this now. It's got 4 or 5 years. So what are the hurdles? What's the holdup, Mike? I mean, Maybe I'm.
Well, I think as I said earlier, and you know, the early hurdles of regulatory uncertainty are behind us for the most part. I mean, there's still some areas, as Hillel noted, but for the most part, FDA has put forth a lot of good clarifying documents and corrected some earlier ones that weren't quite so good at what they described. That being said, it was an ongoing learning process for them and for the industry too. So I think that's sort of behind us, and now we're going forward with these try to overcome these commercial and patent type of legal type of hurdles, and I think that joint collaboration between FDA and FTC will certainly help. On the technical side, there's still the challenge for more complex molecules, and I think we're going to talk about those later. But those are those that are beyond monoclonal antibodies, which are pretty well defined, quite stable for proteins. There's a lot of other proteins that don't fall in that category, and they present more challenges.
Okay.
You know, maybe I can ask a question to both Mike and to Rick. In order for biosimilars to be profitable, you have to have a big, wide margin between the cost of goods and what you're able to sell it for. As biosimilars get used more and more, the prices will inevitably drift lower, hopefully just drift lower as opposed to falling like a rock. We don't see that happen like you do with generics. But there's also a need to lower the cost of goods, which is, I think, your areas of expertise. Is that something that companies are looking at?
Definitely are looking at it. Hello, Mike, you okay if I take this?
No, go ahead and start. I can add on, sure.
Yeah, definitely something they're looking at. They're looking at the processes where they can bring down costs, make sure batches are consistent from one to the other. So there is a drive to get more yield out of each run, batch-to-batch consistency, quality by design. So all of those build into that. I wouldn't say it's perfect at the moment, but we're moving in the right direction. And of course, cost of product, it's never good enough, yeah? When you get there, next goal comes along. So that's what we're working on with a number of customers.
Yeah, I could add there's no question that biologics are much more expensive to produce than small molecule drugs, and that you have a lot more complexity and hurdles associated. It's a longer time span, but that time from when the cells are first thawed to when you have product has been shrinking dramatically to a number of innovations. And for example, the very simple one that a lot of companies are doing is rather than starting with a small number of cells that are frozen, you freeze 10 times as many cells. It's still a fairly small container, and you start with that and you save several days on your process train. Other ones, I think, you have in the area of trying to make processes more efficient in terms of streamlining them, and getting rid of hold times is more almost like something out of Toyota's handbook or something, where you're getting rid of dead time essentially by doing continuous processing. And this has been a sort of a hot area in bioprocessing for the last several years, and we've seen, especially Rick's company, come forward with some very novel products to help that, enable that, combining ultrafiltration, diafiltration in one step, for example, and doing some other tricks that allow you to, instead of doing 2 steps, you do 1 step. And so it's the ideal to go from, the cells all the way to the finished bulk substance is still a little ways off, I think, for most products, but we're getting there by, instead of 10 steps, it's 8 steps, or it's 6 steps, or by combining steps, and more powerful chromatography systems that allow not just separation by one mechanism, for example, hydrophobic interaction, but also by ion exchange at the same time. And so this is called a multimode type of separation, and this allows you to bypass steps too. So there's been a lot of this focus to kind of streamline it and get a faster process, because time and plant is a lot of money. And so the faster you can do something, the more lots you can produce per year, the better. And the other big thing that's been going is to get away from big stainless steel tanks and get into single-use systems with rapid turnaround and allow, again, more processing per unit time.
Is that a two-edged sword? I mean, single-use systems, I was told, and I'm not an expert on like the two of you are, that it's also more expensive per dose to use it, but although it's more flexible.
Well, yeah.
That's changing, I think. That probably was correct a while back, but with the advent of more efficient processes, shrinking the footprint, definitely single-use kits, so we're able to combine a number of things and reduce the costs. So, and that goes for the industry as a whole, this company I work for. So reduce costs for those kits, they clip in place, brings efficiencies. So for the overall dollar per gram, it has less impact. Also another thing we're working on is not just us, but again, the industry is more efficient use of resins, especially protein A resin, things like that. If you can get more out of that, obviously help lower the costs. And then switching some technologies, not all, from resin to fiber-based chromatography as well, just for the speed of throughput and processing.
Can we also talk a little bit about the analytics as well? Well, two things jump out at me. One, and we haven't talked in previous years, but this past year, we've seen an increasing number of situations where we found that reference product itself has changed over time. It's actually making it more challenging for the biosimilar company because now we're moving target. In fact, just recently was one. And the other thing about that is, from your perspectives, multimode analytical methodology, especially mass spec, where you can get a lot of information, more information than we've had previously, and with single assay can we give you multiple assays that previously, so you can speed up your analytics and actually make it more powerful at the same time. Maybe you can speak to those.
Yeah, you can go
ahead, Mike. You take it.
Okay, you can do a lot more assays for a number of reasons. One, each assay is faster. They're often multi-mode, as you alluded to, so you're getting essentially two orthogonal assays in one shot. And then you're using much less sample volume, whereas we used to use milliliter, now you're using microliter quantities. And so you're able on these micro plates. When I was in the lab years ago, we had a 96 well plate. Now they have 4 times as many wells with 384. And I think I even saw a reference to 1,024 or something like that number of wells. It's phenomenal, the miniaturization and the speed at which some of these automated systems, I should also mention automation, has added a lot to allow you, because one of the things that you hit when you have all these assays going so quickly is enormous amounts of data. So you need to have high throughput data analytic capabilities and really terabyte storage capabilities, because you'll quickly get inundated by the amount of data. So you really need to be able to process that very rapidly and eliminate, what you don't want, and keep the relevant data and process that and go forward, instead of years ago one laboriously transferred data into Excel spreadsheets and so forth. Now that's passé and not done. You've got it all automated directly in the instrumentation, and that's added a lot of power to the technique, and also reduced errors, because when you transfer data from one program to the next, you can have human error. And,
so this is true for biologics. This is true for biologics as a whole, but you're saying it's more. It's probably more important for biosimilar development.
Well, because biosimilars have such a heavy analytical burden, Hillel, they really require a lot more analytical expertise and comparability constant to the reference product than you get with if you're an innovator of a new product, then all the data that you're comparing against is your own, and you don't have a set criteria that are all ranges defined by a reference product. You're finding the quality attributes and their ranges with a new product. With a biosimilar, those are largely defined by the reference product, and you must fall within these ranges as defined recently in the May 19th last year, the guidance, May 2019, guidance on analytical. It's very valuable guidance, and it's the FDA said for the first time some of the things that people were wondering about, well, how many lots of reference product do you need? And they said at least 10, but most people were using more than that, and depending on the size of the market and how many lots are available, they may well expect more than 10. And they also had other things like defining the range of that you can work in, and why you should target the midpoint of the range, and some other things that are sensible but were never really spelled out before. So I think that made it much clearer.
Put the two things that I said and what you said together, you'll realize why people are using many more lots. If we're now finding so many more variability in the reference product itself, if you buy 2 or 3 lots in year 1, in year 5, you find out that the reference product has changed. By the time you want to get to the market, then your comparable product isn't really comparable anymore. The more samples, the more variability, the wider and the more accurate your range that you should be shooting, that you have to shoot for.
Yeah. The FDA cautions against targeting the edge of the range because you may get out of it. And you can't really widen it by looking at other sources, other countries, and so forth. That's not allowed either. You have to take a U.S. licensed material.
Yeah, that's actually a bit of a sore point for industry.
Yeah.
Go ahead.
So Hillel and Mike, some of the advances in LC mass spec do give more data in a time period, which allows for more data to be gathered. The MVDA, multivariate data analysis that everyone's doing, or should have in place, is gathering that data, allowing for screening of different spectrum, see where the changes are on a more real-time basis than waiting overnight and all the rest of it. Same goes for ELISAs. They can be analyzed, in a much quicker time period. All of this is leading to more informed decision making. And how real is a change from the actual standard? And what does it mean? Because quite often it's not a significant difference. That can be argued with more data now.
Actually, it's in a way the early stages of development when you're choosing your seed and the right clone. That's normally a very big chunk of time in the development of biosimilar, getting the right product to development, and the ability.
Yep.
And as Mike's saying, the ability to do analysis on much smaller samples and maybe on complex matrices allows you to screen many more clones and many more seed banks than have been previously done. You can actually proceed faster and with a greater degree of certainty than what you've had in the past. So that actually, this technical ability to do these analyses actually speeds up early development quite considerably.
No, I agree completely.
And then the equipment that it has, like the ability to do more than one analysis, so you can get the actual spectrum for the product, regards its conformation, but also then sort of its affinity, hydrophobicity, glycosylation. Get those off one instrument and one sample. That's speeding up things as well. All of those are moving into the lab. Not all of them go into production because you don't want to measure all of those attributes on the product every day or every batch, but definitely allows you to screen the right product as you move forward.
It's a very good point you raised. I mean, when you have multi-mode analyses, you may want to say, look at one parameter, but if your instrument gives you 6 parameters, then you'll of course collect the others as well. So you're actually collecting information now that you may not have collected in the past.
Definitely, and it's making more informed decisions.
Yeah, and it does speed early development, as Hillel points out, because whereas previously you might look at 100 clones, now you can look at thousands. And then if you look at enough clones, you'll find one that is producing continually and reproducibly and over time a product very comparable to the innovator product.
The other area of analytics that is increasing as well, or PAT, is on the process, where we're looking at concentration in line, aggregation, things like that. Again, all allow for improved process, better yield. But again, that's that's all work in motion, and some you can still do offline, but some things are moving online.
But I want to let's take what you just said and a little bit differently. One of the big things about a biosimilar when talking about the different parameters is what's clinically relevant. And those are the ones that you need to focus on. Aggregation being an extremely important one because aggregates are known to be more immunogenic. If you can identify what's clinically relevant and hone in on those early on, not just that you're getting the information, but you're getting the most relevant information. And this is the analytics of today are much better than they were 15 years ago.
Question.
Okay.
So one thing I wanted to this is information, but Rick, let me ask a very simple question. What molecule? I mean, all this is all this comes to play, but what molecules do you see people targeting in the industry now?
Well, okay. MABs, recombinant proteins, and other variants of that. Okay, but they're all there. Particularly in biosimilars, though, it tends to be MAB heavy. And aggregates, as Hillel said, and Mike, is one of the key things that need to be under control, monitored, measured. Definitely something you don't want happening in the process.
Russell, you can ask your question a little bit differently, which would also be interesting for this audience, is what type of products are now being targeted? The initial targets were very large blockbusters. Will we start seeing biosimilars to products that have perhaps fewer sales? So instead of being a $5 million, billion-dollar product that's being targeted, will companies start targeting products that are maybe 500 million or 1.5 billion? So the next generation, yes, there are some blockbusters coming off. When we start seeing biosimilars being developed to smaller targets. And I think the answer to that is relatively straightforward. If the biosimilar pathway is successful, we will start seeing a biosimilar to some of these slightly less lucrative markets compared to the Humira. But if the market is not as successful, then perhaps not.
Yeah, I would agree with Hillel's analysis that we're going to see products in lesser markets or even perhaps some rare diseases where you're not treating a lot of patients, but you're treating them for life and you're continuing every week or month to treat the patient. So it's not so much a cure as a way to manage the disease. And so those kind of treatments stay with the patient throughout his or her lifetime. And I think even though the total number of patients for one of these diseases is low, where there's often just one company producing the therapeutic, like Myozyme or Lumazyme, for example, for Pompe disease. And so there are companies looking at biosimilars of that, even though it's a small market. And some of these enzyme replacement type of drugs, though, are more complicated because they have a lot more carbohydrate. They have a lot more complexity than a monoclonal antibody, and they have some stability issues. So this will present new challenges for the development and manufacture of these type of biosimilars. The monoclonal antibodies are so well characterized now, and they've been so dominant in the industry that they're not so technically challenging as these other proteins.
Just imagine saying that 20 years ago, saying that.
What's that?
Go back 20
for 20 years I've been saying that, right?
Yes.
Well, it's true. They won't ever go away because they're such versatile proteins. And I first studied monoclonal antibodies in 1975, so I've been with them for a long time, and I love the molecules, but I mean, we have to be beyond them.
When we started first talking about the biosimilar pathway in the early 2000s, the line was that monoclonals and EPO were perhaps so complicated no one would ever be able to copy them.
Yep.
And now we're saying they're passé. It's reasonable to project that the analytic advancements and the ability to manufacture a precise product will continue to advance. So things that are challenging today will be less challenging in the future.
Well, we see a building on that, Hillel, is that we see that in Asia now with a lot more activity than say in Europe or North America. with biologicals. And it's mainly, I think, because speed of process, analyticals are allowing understanding of what is equivalent and what can be used as a biological, even for these lesser volume products for critical diseases, but not blockbusters. So there is a lot of activity.
Let me ask you a question then, Rick. For bios, there's an awful lot of companies in India and in China that are racing to develop biosimilars. Not all of them are, some of them, we call them intended copies and not developed to the same level of comparability. Not to say that they're not good products. They could be quite safe, quite efficacious, but they're not really biosimilars. It almost seems that the scientific ability to make these products is there, but to make them as a biosimilar to the standards of Europe and US is not happening as quickly. Is that correct, or am I misreading?
I would say maybe true a while back, but that's changing. And definitely, I'll just say it's changing, and the things are coming through.
Yeah.
Well, we're gonna ask this question then. So if you go by region, Rick, I mean, are we still in a catch-up mode here then? Or?
No, I think, well, US is definitely in a catch-up mode, but it's catching up quickly, well, relatively quickly. The FDA improvements and FTC effects that Russ and Hillel, sorry, Mike and Hillel spoke to are definitely going to help things. And companies are pushing forward with that. But we have got to get litigation under control and false information under control, because it doesn't help. Europe, you know, they have different way of defining a biosimilar, but they have more approved already and will continue along that route. But I see the big wave coming from Asia. And I wouldn't say they're all, you know, not to the same standard. I would say the standards are definitely improving, and those products are definitely on the way, and there's going to be a lot of them.
Yeah, when you're saying I'm more concerned about products being developed in Latin America than necessarily I am in terms of because they have the non-comparable pathways in places like Brazil and Colombia that you don't have elsewhere.
That I have less visibility to today. I could probably dig into that. Not seeing quite the same level of activity.
Let me ask you this then, because, you know, believe it or not, we're coming around to almost having to wrap up here. But what we'll do is,
by quick, Russ.
this is the way every year we start talking about it. I mean, one thing we didn't touch on was interchangeable versus biosimilar. I mean, I want to just throw that out there. That's the first question I wanted to ask. Who wants to? You want to take the lead on that one, Hillel?
I'll take that one. I've been living that for a long time. Well, let's say interchangeability as defined in the US. Interchangeability in Europe simply means the doctor's ability to switch. Interchangeability in the US is a regulatory definition that's actually baked into the BPCAA itself. And that means that the pharmacist can switch a product, can give a biosimilar in place of a reference product. Right now, they can do it already. First of all, let's be clear about this. A doctor can give whatever product they think is appropriate and issue. A pharmacist, if they want to substitute a biosimilar for a reference product that was prescribed, can still do it, but they first have to contact the prescriber. So interchangeability in the US is where the pharmacist has the ability to make that switch without contacting the prescriber. Now, for starters, that obviously only applies to something in a retail pharmacy setting. So something in a doctor's office, a hospital, the entire concept of interchangeability is moot. It's not relevant for those types of products. And there are quite a few products like that. In fact, perhaps more than half for the biologics fall into that category. But the FDA has established a guidance and a requirement for interchangeability. It's actually a very, very high technical barrier. They're asking for multiple PK switch studies. We've been out there, and Sandoz comments were that companies are voting with their dollars and with their programs, and there are very few. There are a grand total of 3 companies that have somehow announced that they're doing interchangeability studies. All the other, and there's 80 in development, 20 developed, they're not doing it. So the criteria that the FDA has set are just, people look at it, say, can we do it or not? Is it worth it or not? And right now, people are saying probably not. The companies are, Boehringer Ingelheim and Pfizer are both doing biosimilar studies for adalimumab, and Nichi-Iko is doing a biosimilar interchangeability study for infliximab, which makes one wonder because that's not a product that's given in a retail pharmacy setting. Be that as it may. Maybe they're looking for a halo effect. We're interchangeable and the other biosimilar infliximabs are not. So right now there's a technical challenge for interchangeability. There are still a few outstanding issues on interchangeability that are not yet defined. We don't know how interchangeables are going to be labeled. We don't know about the promotional requirements for interchangeables. The FDA has to still define certain things that you would have taken for granted, that the ICHQ-5E guidance on comparability applies to interchangeables. They said it does for biosimilars, but they punted on interchangeables. So there are still a few small regulatory aspects of interchangeability that still have to be resolved. But the big factor, I think they'll have to be just honest. The FDA has set an extremely high bar. Companies are looking at it, and they're by and large not doing interchangeability studies. Mm-hmm. So people are saying, if it's interchangeable, then I'll use it. That's another myth. The FDA has been very strong on this point. Interchangeability is a regulatory standard. It is not a different quality standard. A biosimilar on which you've done additional studies to get interchangeables, by definition, hasn't changed. It's the same molecule itself. So the quality hasn't changed. It's additional clinical data that has to be collected. And that's a clear source of misunderstanding of many people. But still, it gets down to the fact that they have to set these requirements, and people are just not doing the studies.
Interesting. Mike, you want to add anything?
Well, I think Hillel gave a pretty thorough answer there. And from what I've seen from different companies, that they just think it's not worth it to pursue interchangeability, as Hillel pointed out. The value compared to the effort is just more than you want.
Unbelievable. All right. Let me ask one more question because we're going to kind of wrap up here. But Rick, all right, we're going to look. We do this at the end of every one of our biosimilar panels. So give me the 10-year outlook. What types of companies are going to be in the game and who ain't? What's the deal?
I think a lot of the issues will get resolved, and there will be more biosimilars coming through. It won't be immediate. Still going to take time. To what Hillel just spoke to, then there's also going to be delays to certain products coming through. But in say 5 years' time, probably 3 times where we are now, probably more, and then probably over that 10 times by 10 years out. Because there's a need for these biosimilars, keep costs down, to help in different parts of the world with the cost of medication. So it's going to happen. It's just going to need a lot of pushing through and a lot of diligence by companies and individuals.
I think one thing that'll happen in 5-10 years time out there is people will definitely become more comfortable using biosimilars right now than they are right now. Physicians and patients. I don't know if they'll ever get to the same level as they are with generics right now, where it's like 80-90% of the market, but people will become definitely more comfortable using that. The need for continual education, healthcare professionals and patients and pharmacists will perhaps be less. The nature of the companies may change as well. You will still have some large companies getting in there because it does take effort to develop these things. But right now for adalimumab, this morning I counted 12 products that are either licensed or in development in the US. I don't think the market's going to sustain 12 products. I think you'll end up with 4 or 5 products per molecule. And when companies realize they're falling very far behind, they'll look at history and they say, so they'll back out of that particular product and may continue to develop a different product itself. So I don't think you'll have as many choices for an individual reference product. You'll still have a good choice of 4 to 6. and people will be more comfortable with them. Perhaps you'll also see what we have seen already is a pattern of companies are now partnering. There is no single company out there anymore that's doing it completely alone. Both Sandoz and Amgen used to be among the last, and both of those are partnered on some products as well now.
Mm-hmm.
So they're going to be, I mean, Rick mentioned a lot of companies in Asia and India developing biosimilars. They're not going to develop a commercial infrastructure in the US. They're going to be good quality products. So I think those products will probably be partnered with American companies or multinationals with a large American presence.
Right.
Yeah, I would agree with what my colleague said, and I'd also add that there was this consolidation trend that I think Hillel alluded to. A lot of the small companies, after they saw how hard it was to make biosimilars, either gave up or sold themselves or became acquired by another company. But there's a few that have really managed to succeed, and especially in the last few years. A little company called Coherus, for example, has done very well with several biosimilars. And still it's a fairly small company. So I think there's room for smaller product companies too. I mentioned also the type of product. I think we're going to see biosimilars for some of the rare disease type of therapeutics because they're rather expensive, and so there's a real need to address this because patients take these drugs their entire life, and it's over the lifetime. It gets to be a big outlay, even though there are not many of them. There are many rare diseases, so I think we're going to start seeing. I think small companies will take the lead on that, and they will eventually have biosimilars for some of these indications, which are currently just the reference products available.
Thanks. I want to thank you, Rick, Hillel, Mike, as usual. Another great session. Appreciate it very much. I mean, we didn't even touch on some of the stuff. I mean, like we all know, CMOs, I mean, that alone would have been another hour's worth of discussion, if not more. So based on this, I want to thank everybody, and I'm going to sign out.